Students will understand two very important concepts in bioinformatics. One is genome and second is genomics. Now the word genome was coined by Hans Winkler in 1920. Now in eukaryotic cell, the genome is present at various places such as nucleus, mitochondria and chloroplast, and accordingly they are called as a nuclear genome, mitochondrial genome or chloroplast genome. In case of prokaryotic cell, it can be called as a prokaryotic genome or simply bacterial genome. Now this genome having the two words here, one is gene and second is ome. So something ending with ome means what? Set of something. So set of genes present on haploid number of chromosome is called as genome. You can also define genome as the total genetic constitution of an organism. So we actually try to understand what is actually written on 46 chromosomes which are present inside the human cells. Now this word genome makes one more word called as genomics. This genomics word was coined by T.H. Roderick in 1956. Now genomics talks about study of genome through mapping, sequencing and analysis. It also talks about the understanding the functions of this genome. So there are four aspects in genomics. One is like the three aspects are mapping, sequencing and analysis, and the fourth aspect is functions. So students, I hope you are clear with the concept like genome and genomics. Now in the next slide we'll understand more such terms so that your concepts will get cleared more. Now students, let us understand few more terms which are used in bioinformatics. Now what is actually bioinformatics? It is a branch of science which deals with use of information technology in biology. Now we have learned so far the terms such as DNA, mRNA, proteins and one more term like metabolites. Now we know that information from DNA is copied to form RNA by transcription and RNA is converted to protein by translation. Now these protein they work on metabolites to form various chemical reactions. Now the set of genes which are present on DNA is called as what? Genome, which we have learned just now. Now the set of transcripts which are present inside the cell is called as transcriptome. Similarly, the set of proteins which are present inside the cell are called as proteome, and the set of metabolites are called as metabolome. So if you look at these four words now, there is a word ending with OME. So this is a suffix which talks about set of something. So the set of genes present inside the cell is called as genome. Set of transcript is transcriptome. Similarly, proteome and metabolome. Now study of genome is what called as genomics. Similarly, the study of transcripts is called as transcriptomics, and study of proteins is called as proteomics. So in these three words there is a suffix omics which is coming. So genomics means study of genome, transcriptomics means study of transcripts, that is messenger RNAs, and study of protein is what called as proteomics. So students, once you know the basic terms like DNA, RNA, proteins, you can extrapolate that information to the word like genome and all these things, and then study of something is what called as genomics, transcriptomics and proteomics. So I hope you are clear with these concepts. Students, we can broadly divide genomics into two categories: structural genomics and functional genomics. In structural genomics, we try to understand the actual structure of the genome through mapping, sequencing and analysis. And in case of functional genomics, we try to understand what are the functions of the genes and their expression in an organism. Now we can use various technologies like microarray technology to understand the gene expression. Now students, there are following applications of genomics. Now we can use genomics in agriculture to develop transgenic crops having more desirable characters. For example, we can produce a rice variety with vitamin A. So normally vitamin A is not present in rice, so we can introduce vitamin A gene into the rice so that we can get say improved variety of the rice, that is what called as transgenic crops. Now we can use genomics to create genetic markers which can be used in forensic analysis, like say murder cases or other examples. We can also use genomics to introduce new genes in microorganisms to produce say enzymes or some proteins for us. Now, for example, you can insert insulin gene into the bacteria so that bacteria can produce insulin hormone for us. That is what the good example of genomics. Students, I hope you are clear with the concepts like genome and genomics. At the end, I request you all to like my channel, share it and subscribe it. And thank you for watching this video.